So the last chapter we covered was on the evolution of sex. Very interesting stuff. Now we're moving on to chapter 13, which is the origin of species. So what we're covering here are topics related to what a species is, which has been a highly controversial topic in biology for decades, how species form, and the rate of speciation, how fast species form, and some other topics. So the introduction to the chapter talks about these beautiful bears, polar bears and brown bears or grizzly bears. So these two are sister taxa, very closely related to one another. And of course, polar bears are iconic animals. Everybody is familiar with them. They seem kind of cute in a way. Of course, they are deadly to humans. But they're remarkably adapted, uniquely adapted to their Arctic environment. And not just that, not just to the extreme cold, but also to a marine environment. DNA sequencing suggests that brown bears and polar bears began to diverge from one another or split from one another from a common ancestor about 500,000 years ago, 500,000 years ago. Essentially polar bears splitting off from ancestral brown bears. Now, interestingly, they retain the ability to interbreed, to produce hybrids. So a common species definition, the biological species definition that many students learn, states that species are not able to interbreed. And yet these two are considered by biologists to be different species. So for that reason, among other reasons, these two guys provide a great example, introductory example for the topic of speciation, what a species is, how species form, etc. So in fact, these are, I've mentioned, and they're listed at the bottom of our slide here, some of the main topics. How do species originate? And how do they retain their genetic distinctiveness? What keeps them from mixing together and becoming one thing? So these are very important questions because they help us to understand our patterns of evolution. They help us to formulate decisions about the conservation of life's diversity. They are important questions because of topics of disease, of being able to identify what different species are and how they relate to disease. So this whole topic of what a species is and where species come from is super important to our whole comprehensive topic of evolution. So to continue on here, let's entertain this question, what is a species? So of course you probably remember who invented the system for naming species, giving them binomials based in Latin and Greek. Good old Linnaeus, back in the 1700s. Now of course he explained their origins as coming from where? A divine being, special creation, intelligent design, whatever you want to call it. So that was his frame of thought. That's the line of thinking that he had. He was trying to identify and delineate the different kinds of things that God had created. And of course, he viewed them as unchanging, as being sort of fixed on that ladder of life. And he was categorizing things. So he viewed species as being categories. The smallest category in his taxonomic system, with the bigger categories being genus and family and class and order and so on. So taxonomists have always had trouble drawing the lines between species, deciding what species are. Not just deciding what they are, like a definition, but more to the point, who they are. So we deal with this all the time here in North America. Your textbook mentions an example of a type of bird, I believe it was, that existed in England and Finland and Norway, and there were subtle differences between the birds living in these areas. Not super distinctive differences. So there was confusion and debate about, are they different species or aren't they? Now, just to kind of bounce out of this for a moment and sort of draw on what we've talked about and where we've come from in this class, you probably would understand that these lines are difficult to draw oftentimes because of gene flow, because there's geographic variation across space, across geography, due to different environments, different climates, whatever the case may be. And so we see differences in populations across geographic space, and sometimes we're tempted to call those different populations different species. It's hard to draw the line of who the species are. And of course, the next point being made here is hybridization occurs from time to time. And when hybridization occurs, that also kind of blurs the distinction of who's a species and who isn't. So that's a challenge. And I think I've already mentioned the point, the first, without really realizing it and seeing that it's my second asterisk here, is that there's large amounts of phenotypic and genetic variation that occurs within species, and so there's differences across geographic space. So that's what I was talking about on the first point. Your textbook has this quote from Charles Darwin, and I think it's a good one. He said, he's a sharp guy. He said, it's really laughable to see what different ideas are prominent in various naturalist minds when they speak of species. It all comes, I believe, from trying to define the indefinable. So apparently Darwin understood that species are not easy things for us to grasp, not easy things for us to delineate so easily. I think this passage, I think this quote gets that across. Darwin was way ahead of his time. So which evolutionary forces are responsible for transforming independent lineages in their separate environments? So just draw back on those main mechanisms that we have talked about. Think about it. Which mechanisms, which forces would cause independent lineages to change, to evolve, to transform, when those lineages are in separate environments? Of course, the weakest force would be mutation. It just supplies a new variation that wasn't there before. What are the probably two strongest forces? Especially if a population is very small, either because of a founder effect or a bottleneck, that would be genetic drift. So that would be a primary force that would transform or change independent lineages. And its counterpart, the one that's really more effective when populations are large, because then genetic drift can't take a grip, that would be natural selection. Those would be the transformative forces. And while yes, gene flow is also a powerful evolutionary force, remember, gene flow doesn't make populations different. It actually keeps them more homogenized and the same. So it's genetic drift and natural selection are the primary ones. And overall, the bottom line message is that when independently evolving lineages, groups of organisms that we can identify are separate branches in the tree of life, when they have sufficiently diverged, when they have become different enough such that we can recognize them and tell that these lineages have been evolving separately, then we call them separate species. And as you'll see coming up next, as I discuss this some more, that's really what we're doing, and that's really the criterion that we use. Are populations evolving independently from one another? Have they been? Can we show that they are distinctive lineages? Then we might call them separate species. So let's keep going with what is a species and dealing with some definitions here especially. So amazingly enough, there has been about 25 different species definitions. And of course, these exist through publications, primary journals, books, where biologists have made arguments of what they think a species is. And this really began, I think, back in the 1940s with the biological species concept or definition that Ernst Mayr published and proposed. Very famous evolutionary biologist of the 20th century. You should look him up. He was an ornithologist and widely regarded as sort of the father of speciation. So in the 1960s, there was a definition of species that came to compete with Ernst Mayr's definition. I believe it came from George Gaylord Simpson, who was a mammologist, paleontologist, systematist, and he argued that the biological species definition really didn't reflect how we understand that evolution occurs. It wasn't really consistent with evolutionary theory. So he came up with a definition that we call the evolutionary species concept. And going into the 1970s, then there was a real flurry of debate as people became really interested in this subject. And it really did come down to differences in philosophy, as I mentioned here, but also one's particular biological background, bias, if you will, on how people viewed biology. So, for example, ecologists came up with definitions that focused on ecology and that species sort of have different ecologies. That was called an ecological species concept. Geneticists, kind of thought about things differently, and at some point, I think it was in the 1980s or so, there was a genealogical species concept. So there's all these different ideas. And again, differences in philosophy, differences in background, differences in research methods, etc. But as your book focuses on three concepts. And fair enough. I think I'm okay with this. So the first one that they talk about is the phylogenetic species concept. And very useful to systematists, very useful to those people who study the tree of life, phylogeny, evolutionary relationships, because it focuses on the phylogenetic history of organisms. And basically regards species as being the tips of a phylogenetic tree, the tiny little branches on the phylogenetic tree. And it basically defines species as being independently evolving lineages. That's kind of what the evolutionary species concept said, that are diagnosable, meaning we're able to find evidence to identify them. Phenotypic characteristics, genetic characteristics, ecological characteristics, something that we can use to say these are separate branches in the tree of life. They're evolving separately. So the biological species concept is the other one that's mentioned. Ernst Mayr, 1942. And this is the one that many textbooks, but in beginning textbooks in biology, give as a definition. And actually or potentially interbreeding populations which are reproductively isolated from other such groups. It's the one that you probably learned about in Bio 182. And depending on your professor, they may have talked about other species concepts. I generally talk about the general lineage concept as being the most favorable one, and we'll get to that one on the bottom. So the biological species concept focuses on the process by which species form. That's what your textbook says. I kind of scratched my head about that, and I thought to myself, to me, and not that I'm the big authority here, like I know more than the authors do of the textbook, but I am an evolutionary biologist that focused on speciation in the past. I think the biological species concept focuses more on what keeps species separate from one another, what keeps them isolated from one another, which is reproduction. That is the criterion. They're reproductively isolated. But I think what the authors are saying is that by focusing on reproductive isolation, that we can then investigate and look for the reasons that species are reproductively isolated, and maybe getting into the genetics of what makes them reproductively isolated, which would involve potentially process. So there's that. But the downfall of the biological species concept is that it's only applicable to sexually reproducing organisms. Look up there at the definition. Population, or actually or potentially interbreeding. Okay, well, you can only interbreed if you're a sexually reproducing organism. But there are a lot of creatures on Earth that are asexual. Not a lot of animals, but some are. There are asexual lizards, there are asexual fish, there are many asexually reproducing invertebrates. Many species of plants are able to reproduce asexually. Lots of protists reproduce asexually. The majority of them do. Parameciums, amoebas, and of course, bacteria and archaeans reproduce asexually by binary fission. So this is one of the big criticisms of Ernst Mayr's definition, is that it's really leaving out a lot of different kinds of life forms. And then the final point I make here with my asterisks is that it's only practical when it's possible to study the extent to which populations can interbreed. There's a lot of populations out there that are geographically isolated, like, say, populations of lizards living on different mountains, that you might be able to see the mountains as a human geographically across the landscape, but these lizards are trapped on different mountains. They can't get across a valley in between. So it's not possible to study the extent to which those hypothetical lizards could interbreed. They're stuck on those different mountains. So these are real shortfalls. And so Kevin DeCaros in 2005 first published this concept that he called the general lineage concept, in which you can see here their species are considered metapopulations of organisms. So that means like different populations that are connected to each other, potentially genetically, or have a recent history of being genetically connected. And they're evolving independently from other such populations. So our evolution textbook seems to identify this one as being the one that evolutionary biologists have recently gone along with. And it's very appealing to me. If you read Kevin DeCaros's work, he basically points out that all of these species concepts that people have argued for and published all have one thing in common. They're attempting to identify different lineages of life, whether sexual, asexual, plant, animal, or whatever. But they're trying to identify these different lineages in different ways. So one way is through reproductive isolation. Another way is through looking at the genetics to see to the extent to which populations are genetically different. So that's really, he argued, what the differences fundamentally are that people have been bickering about for decades, is how we identify lineages as being different, or at what point we're evolving that, identifying them to be different. So at this point in time, the debate has settled down quite a bit, and it looks as if the general lineage concept is the one that most evolutionary biologists have settled in on. So as I was saying on the last slide, and of course continuing here with this video, the general lineage concept is arguably the most useful one. And because it really kind of unites all the different concepts in saying that we're all basically identifying lineages as being different species, but we're just using different criteria to identify them. So for studying how new species form, again, the authors of our textbook are saying the biological species concept is very useful. And as I mentioned a little bit earlier in this narration, that's likely because reproduction is central to many kinds of plants and animals, sexual reproduction. And so that provides a framework for us to understand by understanding how different lineages become reproductively isolated. That gets down into the process, of how it happened. I don't know. I mean, again, this is leaving out asexual creatures. So for sexually reproducing species to remain distinct and not, genetically unite into one thing, there has to be an isolating barrier to gene flow. There has to be. Otherwise, different populations will meld together by gene flow and become one cohesive, homogenized group of organisms over generations. So what are the isolating barriers? What could they be? Well, they could be extrinsic. That means it doesn't have anything to do with the organisms themselves. So geography is the most likely one. So if populations are simply isolated geographically in different areas, then that will isolate them from being able to reproduce, just plain and simple. But that, again, that has nothing to do with the biology of the organisms. It has to do with the fact that they live in different places and they can't get to each other. Or they can be intrinsic barriers. And there are lots of those, as you will see in a few moments. They can be intrinsic barriers that minimize interbreeding or completely prevent it. And regardless, populations could be separated by either or both of these. They could be separated by only geography or only intrinsic barriers, or potentially both. Populations could be geographically isolated, but we could also discover that they have evolved intrinsic differences, like different breeding seasons or different mating behaviors, such that both of those things, extrinsic, intrinsic, and intrinsic, cause them to be isolated. So there's a figure here. It's a nice little figure. It's a little bit different from what your textbook shows, but showing basically this idea that you can have lineages within the original species, basically populations that are forming a meta-population. And there's different levels of gene exchange or gene flow between these lineages. But when a barrier to reproduction emerges, so there's this little block over here, then that barrier could be extrinsic, could be intrinsic, but when there is a barrier that arises, then we're going to get these lineages splitting up and becoming independent from one another, and they're going to start diverging. They're going to start changing due to mutation, genetic drift, natural selection, sexual selection. And at some point, we might be able to identify them as having changed enough that we go, oh, they're different species, they're different lineages. So that's the concept. So when it comes to geographic barriers, this example is out of your textbook. There's gazillions of examples, but it's a great one. So we've got these animals that live here in North America that we call elk, and we have these animals that live in Europe and Asia, Eurasia, that we call, I guess, red deer. And we regard them as different species. And these different groups are allopatric. So that is the most common factor that prevents interbreeding, which is allopatry. That's just a fancy word for meaning geographically separated. So right now, the rest of my family is in Northern California, so I am allopatric with respect to the rest of my family. We are geographically separated. So we've got these elk, as I said, of North America. They're called something different in Europe and Russia. And they share a common ancestor going all the way back about 7 million years ago, being widely dispersed. But about 9,000 years ago, rising sea levels isolated them. Sea levels began to rise a little bit before that, but they had risen enough such that they couldn't get across the Bering Strait anymore from, up here to over here. And so with them becoming geographically isolated from those rising sea levels, now they are truly allopatric. But 9,000 years is not enough time for animals like this that do not have a lot of offspring per reproductive effort, and who have relatively long generation times, not on the order of days or weeks or months, but years perhaps, for intrinsic reproductive isolation to have evolved. 9,000 years is not enough time. So if we put them together, we take an elk from North America, and we'll just say an elk from Europe. We put them together in the same place artificially. They can reproduce, and they do just fine. They're offspring. So they don't have intrinsic isolation. Most species concepts, the final thought on here, species have to remain cohesive even when they're in sympatry. And so if these elk were to become sympatric, ocean levels are rising because of climate change that we're causing. I don't know if they will rise enough to connect, Asia with North America. That would take quite a bit of a rise. But if they do, then these elk will be able to go back and forth again, and they will become sympatric. But for now, and for the past 9,000 years, they have not. So let's take a look at some of these reproductive barriers. Finding a mate is what this is titled. And there's a lot of different reproductive barriers, as you'll see shortly with a figure from your book. It goes into quite a bit of detail. So habitat can isolate. So many species of fruit flies, for example, simply lay eggs on different fruits. So these fruit flies can be flying around in the same area, in the same field, the same meadow, whatever, same forest. But if they are adapted and have evolved to be laying their eggs on different fruits of different plants for a variety of reasons, then those habitats will isolate them and they will not reproduce. So that's one. Timing can isolate. temporal isolation. So it can be pretty incredible dramatic timing. Like your book has this fantastic, spectacular example of sympatric species of corals living in the ocean. So these coral species live in the same areas on the same coral reefs, but they release their gametes at different times during the day, sometimes only separated by 1.5 to 3 hour differences. See the figure down below, the sort of bottom left. Spawning times. So one species might be releasing its eggs and sperm in the early to mid-afternoon and then stop. And then those gametes are drifting around and swarming around in the ocean, and fertilization is potentially happening. And then another species in the same area, sympatrically, starts releasing its gametes in the late afternoon to early evening. And again, the gametes are drifting, the currents are taking them to other places, and so there's a narrow window of time by which those gametes can be combining. It can be that slight of a temporal difference. So it's a pretty dramatic example, but it's real. The other example here are these two species of monkey flowers, which belong in the genus Mimulus. And they're partially sympatric, so most of their ranges, the populations live in different areas, but in some areas they overlap and you can find the same or these different species of Mimulus living in the same area. But each is adapted to different pollinators. Your book is very interesting to read about this, the details at which they're different. So for example, one of the flowers, Mimulus lewisi, is adapted to having bees pollinate it. And so the shape and structure of the flowers, even the color of the flowers, is matched to, optimized in a sense, to bees pollinating. But the other species, again, these are partially sympatric, Mimulus cardinalis, is matched to hummingbird pollinators. So the shape of the flower is different, whereas the bee flowers are offering more pollen, which is what bees are looking for. The red flowers, the Mimulus cardinalis, that are pollinated by hummingbirds, offer more nectar, because nectar is what hummingbirds are after. And so there's these differences between them. And these two species of monkey flower really do not hybridize very easily or at all because of different pollinators. Pretty wild. They can be easily hybridized by hand. So we can take pollen from one flower, like a Mimulus lewisi, and put it on the stigma of a Mimulus cardinalis, and they'll reproduce. But the point is that naturally it doesn't really happen. So this is highlighting again habitat and time. So what we're looking at here, and your book goes through these many examples, is different ways that there are reproductive barriers to forming a zygote. Because that's what it all comes down to, is, well, at least with sexually reproducing organisms, of course, and that's the main focus of what's being talked about here. So, for example, these species of beetles, Carabus myosansis, and I don't even know how to say that, Carabus iwawakaniensis, try that again, live in sympatry in Japan. And what's going on there is that the male genitalia are different. And so if the male of one species of these are beetles, attempts to mate with a female of the other one, the genitalia can break off within the female and basically kill her. Well, so much for forming a zygote. So, that would be mechanical isolation. You might be familiar with that term. Or the next example, complex courtship displays can also prevent mating. This is very common in insects. But there's also courtship displays in birds and salamanders and fish, little dances they do. If the wrong display is given by a male, most of the time, the female often will not fertilize her eggs with his sperm. So that is most common in insects. So the male might actually deliver his sperm to the female, but she then has physiological blocking mechanisms that prevent the sperm from fertilizing her eggs. Pretty wild. Other incompatibilities involve gametic isolation specifically, such that the sperm might come into contact with an egg, but the sperm can't fertilize the egg. So within a female's reproductive tract, she might have ways of preventing the gametes from combining, or it might come down to binding proteins on the sperm and the egg. Remember, there's receptors on the plasma membranes of the sperm and the egg, and these have to match up with one another. If they don't match up, then spermie is not going to get its nucleus into eggy. So those binding proteins might isolate them. And finally, viable offspring. So there's this Bateson-Dobzhansky-Mueller genetic incompatibility is what it's called. And this idea was formulated, almost 100 years ago. And it was a theory that there's genetic incompatibilities that are the cause of sterility in hybrids. So two organisms just may be genetically incompatible. They belong to lineages, to kind of put it in the terms that we're talking about in this chapter. They belong to lineages that have diverged and evolved independently to the extent that they now are genetically different at key loci, and those genetic incompatibilities cause hybrids to be sterile. And that's what's being highlighted in the figure that I inserted on this slide. It's talking about genetic incompatibilities between these two species of mimulus, monkey flowers. And what's also highlighted on here is that researchers used QTL, quantitative trait locus analysis, to identify where in the genome the loci exist for the genetic incompatibility. So figure A is showing that they've pinpointed it down to a position on chromosome 13. And then figure B is pointing out just how the alleles that are found at that position lead to this genetic incompatibility. So check that out in your textbook. So I've been kind of baiting you so far, telling you that there's so many different barriers to reproduction, barriers to gene flow for those organisms that reproduce sexually. And indeed, in Bio 182 here at GCC, in our Campbell biology textbook, it presents these barriers in a more simplistic way. So fundamentally, as you might recall, we've got pre-zygotic barriers. Those are mechanisms that prevent a male and a female from even attempting to mate, or even more specifically, barriers that prevent a zygote from forming. Hence, that's why they're called pre-zygotic. And then there are barriers that exist that are post-zygotic, which means that a male and female attempted to mate and a zygote formed, but then once that zygote formed, something happens afterward that basically keeps these males and females from having successful offspring and thereby contributing to a lineage, to new generations of organisms, mixing their genetics. There's a reproductive barrier. So I don't want to go through every single detail of this figure. Check it out carefully. Pay attention to my study guide for what I want you to know. But as you can see, following the flow chart, the continuum from over on the left, where it says, find a mate, recognize a mate, these would be pre-mating barriers. And you can see how many there are. These are isolating barriers. I'm just reading this, that impede gene flow before sperm or pollen could even be transferred to other species. So we have behavioral isolation, like different mating rituals, ecological isolation, so having to do with species being adapted to different environments, mechanical isolation, so literally the genitalia don't match up, whether it be with flowers, with pollinating structures, or animals, a penis, vagina. And then you can see the diagram continues with ecological isolation and shows you 3 different breakdowns there. Ecologically, there could be habitat isolation. So even though these species might be living in the same area, they live in different bushes, different trees, whatever. Temporal isolation, timing of reproduction. Pollinator isolation, okay? So then if we go back up to this continuum and say, okay, they are attempting to mate, all right, then there's going to be things like post-mating, still pre-zygotic. So copulatory behavior, gametic isolation, on and on and on. Again, I said I wasn't going to go through the whole thing. But then you get to the zygote, and perhaps a male and a female make that zygote, but then you've got your post-zygotic barriers. Could be hybrids are inviable, hybrids are sterile. There's a number of different things. So it's ends up being pretty diverse and complex as to what nature, what life basically allows and doesn't allow in terms of reproduction. And lots of possibilities that can ultimately make organisms reproductively isolated, focusing on that, for sexually reproducing species. Okay, so let's switch now to models of speciation. This, in fact, is section 13.3 in your textbook. And what I'm going to do here is talk about these models and then probably work a little bit into testing these models. And we'll end with this as a part 1. And I'll talk about the remaining sections coming up soon. So models of speciation. Fundamentally, in a very general way, we can identify 4 stages to speciation. And this is theoretical, okay? So like logically, this is what we'd expect. And then we can look to nature and see that, yes, this is very often the case. Put it into practice. Make it operational, if you will. So we start off stage 1. There's continuous variation within the species. You can identify populations going across a landscape. There are no barriers to reproduction whatsoever. And there might be differences among organisms as we go across the landscape, but again, there's no barriers to reproduction. Stage 2, there's some geographic or minor reproductive barrier that comes about. Right? That's the initial stages of separation. And in the figure that's being shown here, maybe a river forms, and the river is dividing up these populations of beetles, insects, whatever. And so now they're separated on different sides of the river. And due to the biology of these insects, they really can't get across that river very easily, so they are more or less isolated. Offspring on each side tend to mate within their groups. So beetles that are born over here are mating with beetles over here, and beetles born on the other side are mating with beetles born on that side. But as time goes on, generations pass, then stage number 3 comes along and barriers strengthen. They get stronger. And groups become more genetically discontinuous. We start seeing more discrete differences. And so if you look at the figure, how it continues, and you compare the insects that are now found on each side of this river, they're becoming more diagnosable. We're able to characterize them and know which side they came from because we see phenotypic differences. We might be able to identify genetic differences. So yes, barriers are strengthening. As they become more genetically different, they would have a more difficult time interbreeding. The final stage, stage 4, is absolute complete reproductive isolation. And at that point, if the river goes away, if the river dries up, something happens like that, and the beetles can now come back into contact with one another, they're able to move back and forth and intermix, they can't reproduce. And why can't they reproduce? Go back one slide and look at all those possibilities. Maybe it's prezygotic, maybe it's postzygotic. If it's prezygotic, maybe it's genetic, maybe it's temporal, maybe it's ecological. There's all kinds of possibilities, and there could be multiple of them involved just in this pair of species. And so this is called allopatric speciation, meaning different fatherland. Allo means different or other. Patric means like patriarchy, father. So this is referencing the fact that if populations become geographically isolated, separate from one another, and they accumulate genetic differences over time, then that is allopatric speciation. And as mentioned earlier, genetic drift and natural selection are the primary mechanisms that result in these accumulating differences. Okay, so under models of speciation, there are several different models. Again, this is theoretical, like, okay, this makes sense, this could happen, but it's also operational, which means that we can go out and we can apply these models and find that in nature, in fact, these different models are represented by what actually happens in nature. So let's go through these. So if diverged populations actually come into contact, so diverged populations, think of like the last slide, the beetles that have become gradually were becoming different. Various outcomes could include, well, stage 4, complete reproductive isolation. It's already in place. They can't reproduce, so it's definitive. They are separate lineages. Or perhaps it's more stage 3-ish. Interbreeding occurs, producing hybrids. And then we come down to, well, if hybrids are not selected against, they do just fine, then the two gene pools may fuse together. There's enough gene flow that they fuse together. If hybrids are selected against, they don't do so well compared to offspring from each of the independent lineages reproducing, then prezygotic barriers may be reinforced. That is called reinforcement. So basically what happens in reinforcement is literally that the hybrids are selected against, and those offspring that formed from reproducing males and females that are part of the same lineage, there might be temporal barriers or ecological barriers or behavioral barriers that those males and females had that inclined them to just mate with each other and not with the other species, and that would reinforce the reproductive barrier. Over generations, that reproductive barrier would become stronger and stronger because hybrids are not surviving, but the other offspring are surviving. That's reinforcement. Again, the emphasis of why it's called reinforcement is the reproductive isolation is being reinforced because hybrids are selected against. Okay, moving on to the next one. In the model of sympatric speciation, that means that species are forming not from geographic isolation, That is usually happening because organisms begin to mate non-randomly. So there are some preferences that females have for certain males over others, and over generations, if that preference becomes stronger, then even though there's no geographic isolation, we could end up with two separate lineages developing, two independent lineages developing. Para means next to, next to. So parapatric speciation occurs when there's sort of a partial geographic barrier, maybe a transition ecologically from maybe one kind of forest to another, or slightly one climate to another. Then that transition geographically could reduce gene flow. So it's not a complete geographic barrier, like the organisms absolutely cannot get back and forth, but there's something there that sort of minimizes the chances of going back and forth. Then gene flow could be reduced and populations diverge. Let's say, for example, there's populations down here in the desert of some kind of whatever, grasshopper, lizard, frog, and heading north towards like Black Canyon City, towards Flagstaff. The climate changes, the rainfall patterns, the temperatures change. And so at some point, there might be differences in the environment such that there begins to be reduced gene flow from those more northern populations to these more southern ones, and the populations begin to change, diverge. That's parapatric. Now, if a range is big enough so that populations are widespread over a large geographic area, then gene flow between distant populations is reduced. And let me just pause there, think about those elk that might exist here in North America. They have a pretty broad range. Or the elk living in Europe and Asia, pretty huge range. So it's pretty unlikely that elk living, say, up in Canada are going to mate with elk down here in Arizona, right? How are they going to do that? So gene flow between distant populations is reduced. And if that's the case, then genetic drift in these different areas is going to produce variation between the populations over greater distances. This is called isolation by distance. So genetic drift is going to cause alleles to randomly fluctuate, and the more genetically distant or more geographically distant you are from one another, then the more genetically different you're going to be. Same thing in humans, right? Differences, variations in human whatever, physiology, skin color, whatever, is basically isolation by distance. And then the final example on here, in ecological speciation, just local adaptations may become specialized, and you end up getting less contact between organisms that are adapting, or the populations are adapting to these different ecologies. And as they become adapted to different ecologies, then the hybrids that might form between them have lower fitness. And if that happens, then there might be selection for different mating behaviors, divergence in mating behavior. So what I'm trying to present in this last one is sort of a domino effect. Different populations are found in different ecologies, different environments, and one thing leads to the next. They start to become more specialized, they come into contact less, hybrids don't do as well, mating behaviors change, so that's referred to as ecological speciation. So again, the whole title of this was Models of Speciation. It's basically explaining to you that there are various ways that populations can become different lineages, independently evolving lineages. So here we become operational. I've used this term. We have theoretical. It's like, oh, this makes sense. This is a good idea, but we don't really know. And then we have operational. That means that in practice, we're going out and identifying examples and using our theory and going, hey, yeah, here, this works here, this works here. Here's an example, here's an example. So now we've got a few different examples. So the first one here, this was actually presented in our Campbell biology textbook, splitting an ocean. So the Isthmus of Panama began to rise about 15 million years ago. So before that, North Central America was geographically separate from South America, and there was an open seaway in between. So these land forms were not connected. And any species of shrimp or fish or, whatever, sea slugs were able to go back and forth between these two oceans, the Caribbean Sea, the Pacific Ocean. They could go back and forth. But gradually, these landforms connected, and the Isthmus of Panama formed. Okay, and now the Caribbean Sea is separate from the Pacific Ocean. And when that happened, then species became allopatric. So one population of the shrimp was then isolated in the Caribbean, and the other population was isolated in the Pacific. And those evolved to become sister taxa, sister species. And they did so allopatrically. But there are numerous examples of these pairs of species that evolved as a result of the Isthmus of Panama forming. So here's another pair. This species here, this population here, became isolated from this population over here because of the isthmus. And over the passage of time now, 3 million years, they became different species, sister taxa. There's like 15 species pairs of shrimp that live on either side of the Isthmus of Panama, and they formed, these sister species pairs formed allopatrically. So this is an excellent example of how allopatric speciation resulted in numerous new species over the course of a few million years. So again, putting some of these models into practice operationally, here's an example that's more of a habitat isolation model. So more of an ecological model. So for populations, we're looking at these fruit flies. These fruit flies belong to the genus called Rogalitis, and they lay their eggs on fruit. And the fruits, they lay their eggs on fruit, and the fruits drop to the ground, and that's where the maggots develop. The little larvae develop in the rotting fruits. They turn into pupae and turn into flies. So this is a great example for studying the earliest stages of speciation. And in the earliest stages of speciation, it's the easiest to study the mechanisms of divergence because we can kind of capture it in action. We can see it happening. So these Rogalitis flies are an example of this, capturing speciation in action, in process. So in the northeastern United States, two sympatric forms of these flies exist. One spends its time on hawthorn trees that have kind of apple-like fruits, and the hawthorn trees are native to North America. And the other form of the fly, Rogalitis fly, lives on apple trees, and these apple trees were brought over by Europeans a few hundred years ago. So that's why this is just happening recently, and we are watching it in action. So the hawthorn trees and the apple trees flower at different times. So there we go. We've got some temporal isolation that's helped to isolate them. And if you look at the bottom figure, figure C down here, it's depicting that. Apples are flowering in sort of midsummer, and or that's the emergence, and then hawthorns are later in the summer. So there's some overlap there, but different times. Now, these flies are genetically programmed to metamorphose and turn from larvae into flies according to the fruits. And so, there's 19 species of these trees, and these flies recognize different fruits by their odor. So now apple breeders take advantage of this. There's less competition, there's less parasitism by wasps, etc. So it's kind of a side note. But what this is all showing is that this is an example of sympatric speciation, and ecological speciation. So why is it both? It's sympatric because these different species of flies are forming. Look at the cladogram here, figure B. It's showing these different lineages that are forming on different fruits. And this is all happening in the same geographic area. These are not allopatric. These plants are basically sympatric, but the flies gravitate, as mentioned here, towards specific species. So even though these species of plants and their fruits are in the same area, it's a habitat ecology kind of thing. So that's exactly the other point. Because they are specializing on different ecologies, that is causing them to adapt to each different ecology, different flowering times and fruiting times and so on. And that's what's giving rise to these different species. So here's yet another operational example. These swordtail crickets, titled A Song of Speciation. They're only found on the Hawaiian Islands. Of course, there's lots of unique species on the Hawaiian Islands, just as there are on other islands that have popped up around the world. But 37 species, a lot of species of these swordtail crickets. And interesting thing here is that ecologically, these crickets aren't very different from one another. They are not specializing in different ecologies. But they are different species in the sense that they are different lineages. They're evolving independently, and they're doing this because they are reproductively isolated. So that has turned out. So constructing the phylogeny of these crickets, which you can see over on the left-hand side of the slide, enables us to map out the route that they took from island to island. And this is a good point, this is an important point. In biology, when we take a phylogeny, we figure out how different species are related. And then we overlay that phylogeny on a map, like a geographic area, that helps to show us geographically, make sense of how the species formed. Like, what is it about the geography? What is it about the landscape, etc., that helps to explain how and why these species formed and this phylogeny formed? This is called phylogeography, a very interesting and active area of biology. And what's going on here is that these crickets invaded. Originally, ancestors invaded the Hawaiian Islands very early on when they started to form, about 6 million, not very long ago, a few million years ago. And the oldest island is Kauai. In fact, it's listed here, 5.1 million years old. And then the other Hawaiian islands are successively younger. Oahu is 2.6 million years old. Molokai, part of it, they're about a little less than 2 million years old, etc. The big Hawaiian island is the most recent one, at only about 500,000 years old. And, there's a new island that's forming down here. It's not with us yet, but the geology is still active. Anyway, the oldest lineages, the ones at the bottom of the cladogram, are found on the most westernmost island, the oldest ones. So that makes sense geologically, that this phylogeny, this history of lineages began with the oldest island. Dispersal is pretty rare, so that's another thing that the phylogeny shows us, is that these crickets are not, like easily jumping from island to island, but new species are forming within the islands, on the islands. It's not that dispersal doesn't happen, it's just that it's rare. So that there are adaptive radiations on each island. So, for example, the Big Island, even though it's only 430,000 years old, has 6 species of these sword-tailed crickets. That's fairly rapid speciation. And as I mentioned at the start of this slide, there is no ecological speciation. They're not living differently ecologically, but their songs have diverged. Females prefer songs of certain males. And you can look to your text for this, but I'm just summarizing it here quickly. Again, another example of a QTL analysis being conducted to find the loci and identify the genes that are responsible for this isolation due to song. And QTL analysis has found the loci associated with song production and preference. It happens to be the same genes operating in both males and females that are giving rise to the production of different songs as well as the preference for the different songs. So pretty fascinating stuff. Isn't it amazing what we're able to discover and find out now about the process of speciation, getting down to finding the individual genes that are responsible for the divergence of lineages? All right, well, we're back to the polar bears. So I think this is our last little story, and we'll end here, part 1. But a tale of ecological speciation and interspecies gene flow. So this has turned out to be a pretty interesting story. So let me just go through this with you. So at this point, we now have whole genome sequencing of brown bears and polar bears, and what it reveals is that they began to diverge from one another, the polar bears from the brown bears, between 479,000 and 343-ish million years ago. So that's how long ago they started diverging. And the Earth was much warmer back then. And Greenland was in fact covered by a spruce forest. So Earth was a different place. It wasn't even very long ago, a few 100,000 years ago, but climate was very different. And Earth was going through a warm period. Now, when the climate changed, the northern brown bears became isolated from the southern brown bears, and they began evolving in allopatry, being geographically isolated. And with that geographic isolation, the ecologies were very different. So the climate was changing, it's getting very cold in the north. Of course, it was warmer to the south. And due to this change in climate and change in ecology, there was very rapid ecological separation. It's the best explanation for what has happened. There's very low variation at certain genes within the polar bears. There is very low variation. So what it suggests, drawing on an earlier chapter that we've gone through, is that there was very strong natural selection. And it swept the genes to fixation. And again, that's called a selective sweep. Go back and review that if you don't quite remember, but we can identify these selective sweeps by looking at some key genes in these species. So there's a particular gene, APOB, codes for a protein that binds to cholesterol. And that one turns out to be a very important gene in the evolution of the polar bears, so that they're able to eat a diet that's very high in fat and not suffer health consequences. There's also a very rapid change in hair pigmentation that occurred. and that goes along with the brown bears that were isolated in the north, where there's a lot of snow. Having hair that was white instead of brown would be an incredible advantage, because these bears were feeding more on marine life like seals and sea lions, so being able to blend in with the Arctic landscape would be a huge advantage. These seals and sea lions wouldn't be able to see the bears. There's been occasional gene flow from polar bears to brown bears, and hybridization. And so that's depicted by the arrows that are on the diagram here. So, but despite this hybridization, and they are still able to hybridize, the ecological differences between these bears is great enough that these lineages have remained separate and distinctive, such that we can identify them as separate lineages and say they are different species: polar bear lineage and brown bear lineage. So this brings us through several examples. We have these theoretical models for how species form, and some of these operational examples that I've given through you, and your textbook actually talks about a couple others, helps to give you some meat to put on the bones, if you will, to help to explain and show how new species can form in a variety of different ways, situations, circumstances.